Hello and welcome back. In this video we're going to cover hearing. So we'll go ahead and start with some of the external parts, the middle parts and internal parts of the ear. So if we start on the most external part of the ear, we have the auricle, which is this region here, the outer ear. And what that job of that auricle is to basically harvest light and send it towards the external acoustic meatus. The external acoustic meatus is a passageway through the temporal bone, and it extends all the way down to the tympanic membrane. If we dive a little bit deeper into the middle ear, of course on the outer edge of the middle ear we have the tympanic membrane. This is going to be a membrane that when airborne sounds strike it, it's connected to the malleus and the incus and the stapes. These are our three inner ear bones. These particular bones will vibrate when the tympanic membrane vibrates. And this particular structure is connected to the inner ear at this location right here called the oval window. And so we have some structures over here, the semicircular canals, and then the vestibule, and then the cochlea. So the semicircular canals and the vestibule, they're going to be involved in equilibrium, and then the cochlea over here is going to be dealing with the hearing. So equilibrium is going to be, the information from equilibrium is going to be sent out via the vestibular nerve, and the cochlear nerve is going to send out information about hearing. Of course, together these make up the vestibular cochlear nerve, and then they carry information to the brain. Looking a little bit further in, we do have a cavity in here, the tympanic cavity, and this cavity is going to be drained by this auditory tube, also known as the eustachian tube. Alright, so the inner ear is made up of the semicircular canals. These function in equilibrium, and it's going to be made of the cochlea, which functions in hearing. Both parts are are basically composed of a series of tubes. So you have tube inside of tube. And if we take a look at that tube inside of tube, we do have a bony tube, okay, and then inside there's a membrane that creates an inner chamber. Alright, so the bony labyrinth is made of bone, and then the membranous labyrinth is going to be made of a membrane. Now these two tubes, this tube within a tube, they have different fluids. So the first fluid is called perilymph, and then the most inner fluid is called endolymph. Okay, so we have basically a tube inside of a tube. You can see the bony labyrinth right here in the semicircular canal, and then you have the membranous labyrinth on the inside of that. So we do have two tubes, and these are going to be a tube within a tube. This is it's similar over here in the cochlea, which is the part that deals with hearing. So we do have a bony labyrinth, and then we have a membranous labyrinth. But over here we do have a bony labyrinth here and here, so two bony labyrinths, and then in the middle is going to be the membranous labyrinth. We'll try to show you how that kind of connects together and is going to be involved in our sense of hearing. So let's take a look a little bit more closely at the cochlea. So over here we have the semicircular canals and the vestibule. Those are going to be involved in equilibrium. And then we want to really kind of focus in over here on the cochlea. You could take that cochlea and you could like pull it out, straight out, but you can see that it's coiled over there almost like a snail shell. So again, you could take and stretch this out, and this thing does rotate around and coils around, creating this thing that looks like a snail shell. Okay, so let's kind of take a look inside. I want to take a look and show you kind of how this is organized anatomically inside of each of these parts of the spiral. Okay, so that's what we want to do is go ahead and take a look inside of that. So if we blow that up, this is just one of the coiled tubes. Let me just go ahead and just kind of knock out the anatomy with you. So this is called the scala vestibuli. It's basically a chamber that's going to be full of the perilymph. We then have another chamber that's going to be full of the endolymph. It's called the cochlear duct. And then we have the scala tympani, which is going to be full of the perilymph. Okay, this is the bony labyrinth we were talking about before. This is the membranous labyrinth that's created over here. So we have a series of tubes, tube, tube, tube. And let me just kind of show you how sound's going to be carried through. So sound waves, let me just kind of back up and show you. Sound waves are going to strike the tympanum, the tympanic membrane, and it's going to cause these bones to rock. That's going to take and push fluid that's inside of the cochlea. Okay, so we have the the oval window right here. So this stapes, when it pushes fluid that's inside of the cochlea. Let me show you what fluid that it's going to be pushing. So our initial pushing of fluid is going to be created in the scala vestibuli. And that fluid, when it rocks back and forth, it pushes on this membrane here called the vestibular membrane. That membrane, when it rocks, causes the fluid in the cochlear duct to rock. And when that fluid in the cochlear duct rocks, it creates movement of the basilar membrane. And that basilar membrane is then going to take the pressure waves and transmit it to the scala tympani. Okay, the scala tympani. Now, let me just kind of go back and show you again. So rocking fluid in the scala vestibuli rocks this vestibular membrane. The vestibular membrane is going to rock the fluid in the cochlear duct, which rocks this membrane called the basilar membrane. And when the basilar membrane rocks, it's going to cause these sensory hair cells to basically rock back and forth. And they have little parts that are embedded in this thing called the tectorial membrane. But when these little hairs right here, these sensory hairs rock back and forth, that's going to transmit information back to the brain through the cochlear nerve so that we can actually sense sound. So it's the rocking of fluids in these various chambers that's going to eventually cause the hair cells to rock back and forth, which is going to send information to the brain about what we're hearing. Just to give us a little bit closer view here, so we do have this organ of Corti, which is this whole structure here that you're seeing, and embedded in the organ of Corti are these hair cells that are going to be sensory cells that are going to sense the rocking back and forth. So remember, the basilar membrane, when it rocks back and forth, these hair cells are going to be rocking, and they have little itty-bitty little outer hair sensory fibers that are embedded in this gelatinous membrane. So when these things rock back and forth, these little sensory hair cells embedded in the membrane are going to cause the cell to depolarize and thus send information back through this nerve fiber, to the cochlear nerve. So when we have sounds coming through, so remind you one more time, sound comes through the tympanic membrane. It rocks the malleus, the incus, and the stapes, which basically rocks fluid in this particular chamber right here. So it's called the scala vestibuli or vestibuli. That rocking of the fluid, depending upon the frequency of the sound wave, is going to cause the cochlear duct fluid to basically move, which is going to move that little basilar membrane, and then all the extra pressure waves are going to be dissipated out of the inner ear through this thing called the round window, and it's dissipated into the tympanic membrane. So different frequencies of sound are going to rock different portions of the actual cochlea. So sometimes higher frequency sounds are going to be sensed in one area, and then lower frequency sounds are going to be expressed in another area or sensed in another area. There are some frequencies of sounds that we literally cannot sense, so they go through the fluid, all the way through the fluid, and then they just kind of go into the scala tympani, and then are just dissipated through the round window, and we actually can't even hear those particular frequencies. So we have high frequency sounds are going to be sensed at the very start of the snail, the cochlea, and then mediums frequencies are going to be kind of sensed in the middle, and then super low frequencies are going to be sensed towards the end of the cochlea. Of course, some sounds will not be heard at all, and they'll travel right on around, and then be dissipated by the round window into the tympanic cavity. So just to kind of give you some steps of what we just talked about, sound waves enter the external auditory meatus and they cause the tympanic membrane to vibrate. The malleus, incus, and stapes transmit the vibrations to the oval window. The oval window transmits the sound waves via the perilymph of the scala vestibuli to the vestibular membrane. Sound is transmitted from the vestibular membrane to the endolymph of the cochlear duct. This causes the basilar membrane to vibrate. This vibration causes hair cells in the organ of Corti to move back and forth. Hair cells stimulate nerve cells to basically transmit information to the brain via the cochlear nerve. The leftover sound waves are eventually transmitted to the perilymph of the scala tympani, and this is dissipated into the tympanic cavity via the round window. Just to kind of give you another kind of sense of what's going on here, of course we have vibrations that are being transmitted to membranes, which are transmitted to fluids, which are transmitted to membranes. The rocking back and forth of this basilar membrane causes these little sensory hair cells to send information. Okay, it sends information to the medulla. That's where the vestibular cochlear nerve will innervate, is the medulla. That information crosses over in the medulla, then it's transmitted up through the midbrain and then to the thalamus. The thalamus then sends it over to the temporal lobe, where we're going to actually have the sensation being picked up. Okay, so that's how sound is transmitted and then sent via the nerves to the brain. Okay, in our next little video, we'll be doing dynamic and static equilibrium. So we'll see you then.